Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled A Novel Approach to Removing Urea from Dialysate, Methods and Validation by Amino Acid Quantification. I'm Liam Samuel from the events team here at Scientist.com, and I'll be your host for today's event. This webinar has been sponsored by Biochrome, a division of Harvard Bioscience, so a big thanks to them for helping to make this webinar possible. We're very fortunate to have Dr. Guozheng Shao, acting instructor in the Division of Nephrology at the University of Washington, and Dr. Anna Scott, director of the Biochemical Genetics Laboratory at Seattle Children's Hospital, here with us today, who will discuss the development of an osmotic membrane for the use in a portable dialysis device and its validation using amino acid analysis to quantify urea removal using this novel membrane. Anna, thanks so much for joining us today, and the floor is yours whenever you're ready.
Good morning and greetings here from Seattle, Washington. It's really a pleasure for me to be presenting today. I'm going to do a short introduction, really talking about amino acid quantification and a little bit of context for this collaboration with Dr. Shao, which was really a fun opportunity for my team to branch out from our more typical activities. So I wanted to start with a brief overview of what we normally do, and I think how that makes us potentially excellent collaborators for basic research. So again, my name is Anna Scott. I work in the Clinical Biochemical Genetics Laboratory at Seattle Children's. And from my short introduction here, I hope the audience can learn a little bit about genetic disorders and what medical doctors use amino acid quantification for. I'm going to talk about two different methods that are used to measure free amino acids. And I wanted to talk a little bit about the kinds of assay monitoring and maintenance that a laboratory in a hospital has to perform, and how that can really be a resource to basic researchers, and again, make us excellent collaborators depending on your project and your questions. So briefly, biochemical genetics is a pretty generic term, but what we specialize in the diagnostic world is the identification of inborn errors of metabolism. And these are genetic abnormalities that alter basic metabolism of proteins, lipids, amino acid synthesis or breakdown, sugar metabolism, so glycolysis or gluconeogenesis. Really any enzyme defect in those pathways has the potential to cause some pretty dramatic disturbances. And I wanted to share here the first inborn error of metabolism was first described in 1902, and it's a disorder referred to as alkaptonuria. And it was biochemistry that helped to identify these abnormalities. And in patients with alkaptonuria, their urine, when it oxidizes, will change color and turn, kind of the textbooks describe it as a black color. This was actually a sample in my laboratory that looks a bit like an amber ale in the light as you shift the pH from acidic to basic. So that's really a titration set of samples shown here. But this is a clue that there was something not quite right in the metabolic process, and that individuals with alkaptonuria are excreting compounds that are very atypical. And that's really what biochemical genetics is all about. We do not do DNA sequencing, but we look for small molecules, or we directly measure enzyme activity to find defects in fundamental metabolism and try to pinpoint where the genetic abnormality may be, which makes for much more specific genetic sequencing testing. So inborn errors, as I mentioned, cover a large space chemically. Today I wanted to talk a little bit about amino acids, and there's a lot more than the classic 20 that we think about. I've added some green circles here to highlight some of the amino acids that we are able to measure in body fluids. And again, seeing the accumulation or the absence or even presence of very unusual amino acids can be diagnostic of specific inborn errors of metabolism. So our providers, medical doctors that order free amino acid testing, will typically order it if there's a question of, is there an inborn error of amino acid metabolism, or your patient already has a diagnosis and they're monitoring protein intake, making sure that we're not stressing that particular pathway too much. Sometimes the unusual amino acids that accumulate, or even normal amino acids that accumulate to a level that is way beyond normal, can cause clinical symptoms and actually be quite dangerous to patients. So free amino acid quantification is used primarily to make it a diagnosis or to monitor patients for their management. And in the biochemical genetics lab, we use really any tools that we can to measure small molecules and enzyme activity. So I've pictured here the basic tools that my team uses. A GC mass spectrometer, tandem mass spectrometers with a lot of liquid chromatography on the front end. I'm going to be spending most of today talking about our HPLC system for free amino acid quantification, but we also use UV and fluorescence spectrometers. Again, I mentioned enzyme activity is a big part of what we do, and those are the instruments that we use. So amino acids, this is kind of the relevant component for today. We use an HPLC system that relies on UV absorption to quantify each of the amino acids. We use a derivatization component called ninhydrin that, when it reacts with any free amine, will create this compound called Ruhmann's purple, which is literally purple. And we can, using the UV detector, measure the intensity of that color at a particular point in time that corresponds to each of the amino acids. So that is the system that my laboratory currently uses. Many other laboratories, particularly big reference laboratories that offer biochemical genetic testing, are moving in the direction of tandem mass spectrometry as a method of measuring free amino acids. Tandem mass specs are incredibly powerful and technically challenging. So one of the things I like to tell folks that come through the lab is with great power comes great vulnerability. The mass specs are incredibly powerful devices for measuring specific compounds, but they're not so great at the unexpected. And they are also very technically challenging to maintain, and the initial assay development is also quite challenging. So this is why this particular instrumentation is primarily used in reference laboratories, where they may have entire teams of development scientists able to provide support for this instrumentation. And also they have the sample volumes to justify the initial setup cost and development. The mass specs are quite a bit more expensive to purchase than an HPLC with a UV system. So those are our two methodologies. They each have their limitations and their strengths, and I'm going to show you a little bit of data off of our HPLC system. So the majority of samples that come into my laboratory are plasma samples. So this is spun blood. We use internal standards, and we precipitate out any free proteins in that plasma. And so it's really a supernatant from a quick crash and centrifugation. That supernatant is transferred to a clean vial and loaded onto our analyzer. This analysis process is a little bit on the slower side, but the column takes about 3 hours to run, and it really gives us baseline resolution of each of the known amino acids. An example chromatogram is pictured here with time on the x-axis and absorbance at 570 nanometers on the y-axis. There are ways to speed this up if you don't need all 20, or well, it's like 30 or 40 amino acids that can be detected, but the majority of people have about 25 that are readily detectable. There are ways to speed up the chromatography and target particular sections, and that does have its uses for specific known diagnoses. But generally, it takes about 3 hours start to finish. We run one patient at a time, So this is not the fastest technology, but again, there's a level of robustness here and reliability that's really fantastic. And so I mentioned in the clinical laboratory, we are dictated by concerns of patient safety, which means we spend a lot of effort doing validation of all of our instrumentation and our methods. We worry about accuracy and reproducibility. And so some of these elements are covered by legal requirements. These are dictated by the Clinical Laboratory Improvement Amendments, also known as CLIA certification. Any laboratory that is turning out patient results must be CLIA certified. This is a federal requirement. I would say most hospital laboratories are also CAP accredited. This is an additional level of kind of demonstrating good laboratory practices, again, with an eye towards patient safety and reliability. So the College of American Pathologists is not a legal entity, but they do offer essentially inspection services and accreditation for laboratories performing human testing. And here are just a few parameters that I listed around assay validation. And some of these things we test as a new method or a new test is coming online, and others we monitor regularly. So we do accuracy assessments throughout the year through proficiency testing that's offered by external kind of entities. This is samples that we essentially purchase to test them. We're graded, we're compared to peers, both in terms of methodology and other clinical laboratories. And we regularly monitor for changes in the behavior of our instrumentation. So we run daily control samples, normals and abnormals, to make sure that we are as consistent as possible. And so with all of that as a backdrop, when Dr. Shao reached out to me about his new dialysis device, his question was around really our sensitivity and our accuracy, whether or not we could measure free amino acids in samples kind of before his device and then on the backside of his device, so the dialysate, to get a sense of how many of the amino acids were being pulled out inappropriately. This is a known problem for dialysis in kind of current state. And that's a lot of energy that's missed and removed from a patient when things other than the intended molecule come out in the dialysate. So this is basically my last slide, but I wanted to show you sample one, which was the starting point that Dr. Shao created. It was this artificial mix with a handful of amino acids that are known to readily be removed through current dialysis technology. And sample two, pictured here, is the back end of his system, so the dialysate. So after his device was run for a while, and what you can see is that the peaks kind of pictured, in the interest of zooming, the top and bottom sections are just different points in time. So it's like 0 to 90 minutes on the upper graphs, and then 95 to 135 minutes on the lower graphs. But you can see that those big peaks, particularly in the upper graph for glutamine, glycine, and alanine, are not present in sample two, or very, very minimal. And I was able to show that we were basically at our lower limits of quantification in the dialysate, and that the vast majority of the free amino acids were retained in kind of our blood mimic. So with that as an introduction to the methodology, thank you so much for listening. I'm going to hand this over to Dr. Shao to talk about what he actually did as far as engineering and development. So thank you so much. I will be around for questions at the end.
Fantastic. Well, thanks so much, Anna, for a really great presentation. And with that, I'm very pleased to welcome Dr. Shao. Guozheng, thanks so much for joining us today. The floor is yours whenever you're ready.
Thank you, Leon. Thank you, Anna, for the introduction. I have a sore throat, as you can hear, but I'm not COVID positive. I've self-tested negative on both tests. I'm Guozheng from the University of Washington in Division of Nephrology and the Center for Dialysis Innovation. I'll talk about the portable dialysis we have developed by dialysis regeneration through photooxidation of urea. The problems with the current dialysis treatment is quite obvious. The patients are trapped in treatment centers and they have to go to the center for dialysis 3 times a week. And each time typically lasts for 4 hours and consuming 120 liters of fresh dialysate. The 5-year survival rate for most hemodialysis patients are 35%, which is similar to most forms of cancer. And talk about the 12-hour of weekly treatment. And the patients have very inadequate quality of life. Despite the bad outcome of this treatment is a costly process. Hemodialysis is typically about 1% of the patients and 7% of the Medicare budget in the United States. Since the inception of the dialysis treatment technique, people have been dreaming of portable form of dialysis. On the left is a picture taken in 1974 on the Mediterranean. This is a patient using the READY system. On the right is a more recent photo of Bill Packman rafting down the Grand Canyon in 2006 using Next Stage, as you can see in the middle of the picture. And he was doing hemodialysis. So what we would like to develop is a more portable form of hemodialysis. The advantage of that is quite obvious. The patients will have better quality of life. And presumably, they will operate longer hours, which, from the perspective of uremic toxin removal, will require lower AV fistula or graft blood flow rate, which will alleviate many of the symptoms around the high blood flow rates through the fistula or graft. However, it's technically challenging because we then have to reduce the demanded fresh dialysis from 120 liters in a dialysis session to maybe a few liters. That will make it portable. By doing that, we have to regenerate the spent dialysate and remove the uremic toxins safely and efficiently. At first, we need to understand what are the uremic toxins and how much of the uremic toxins are being removed in each dialysis session. According to the European Uremic Solutes database, there's currently 130 entries. And so what we did was to go over 60 or 70 uremic toxins, and we trying to find out the per session removal as accurately as possible. For example, listed number 1 is urea. The per session removal mass is 23.6 grams. However, most of the uremic toxins are not as well characterized as urea. Then we have to find, for example, the difference between the mean uremic concentration and the normal concentration, and then we need to time that by the volume of distribution. However, although in dialysis treatment, the toxins are removed directly from the vascular space, their distribution, they exist also in the extravascular space, and the distribution is not one-to-one in those different compartments. So we need to define a character called here partition correction, and we try to find as many references as possible to back up data. And what we found is that the previous table being plotted in this figure. Here on the left is the normal concentrations in the serum in milligrams per liter. On bottom axis is the molecular weight. and we plotted the molecular weight cutoff of both the low flux and high flux hemodialyzers. As you can see, most of the uremic toxins, in terms of mass removal, are falling far off the molecular weight cutoff of even the low flux hemodialyzers. And the urea is easily the single most abundant uremic toxin. So we need to have a safe way and efficient way to remove urea. For example, in the READY system developed in the 1970s. And that READY system is centered around the urease enzyme, which converts urea into ammonium ion and bicarbonate. Because ammonium is toxic, then it requires zirconium, the highly specific zirconium phosphate, to remove ammonium ions, which removes other cations as well. It also requires a following zirconium oxide ion exchange layer, which removes the excessive anions. But this READY system gradually phased out when higher water quality control techniques were established in the 1990s. A second method for urea removal is really centered around urea sorbent. And there are dozens of materials being explored as urea sorbent, and many have shown quite high urea binding capacity in the tens of milligrams per gram regime. However, when the urea sorbent, or when the potential urea sorbent exposed to more complicated medium, like a serum, they typically lose their binding capacity very quickly to around 1 or 2 milligrams per gram. Third, another urea removal mechanism is listed in Figure C, where the urea will be oxidized by this oxidation cell, and there will be multiple electrodes, materials and material combinations and operating conditions have been explored. The advantage is that it requires the least maintenance, as it doesn't need the very heavy ion exchange materials, and it doesn't need to daily replace the urea sorbent, which will be many kilograms. And however, the efficiency and safety of this oxidation-based urea method has never been established. So what we decided to go down this path, and we'll do oxidation of urea, and we found highly specific electrodes, which is titanium dioxide coated FTO substrate. FTO is the conductive glass. We found in literature there has been reported 97% selectivity towards urea oxidation on TiO2 substrate. And what we did was to optimize the morphology of this TiO2 FTO substrate. As you can see, in the center image is the SCM image of the nanowire of TiO2 we've grown. And then on the very right, you can see the device assembly. Layer 1 is the LED panel. Layer 2 is the TiO2 coated FTO substrate. Layer 3 is the spacer where the solution is flown. Layer 4 is the air permeable electrode, which will provide oxygen from the air for the oxidation of urea photooxidation. So what happens on the anode is that urea being oxidized into carbon dioxide, nitrogen, and water. On the cathode, the oxygen from the air is used to complete that reaction. So the total reaction becomes urea plus oxygen going completely into carbon dioxide, nitrogen, and water. However, that previous method was not scalable at all, so we developed in-house another method to scale up the production of this device. As you can see in figure A, we have the large LED panel in the back, then large areas of TiO2 coated FTO substrate, and then the spacer for dialysis flow, and we have optimized the flow channel for maximum urea diffusion. And then on the very outer layer is the manifold for solution inlet and outlet. And we tested this device on the benchtop with 1,800 centimeter square of active surface area. We can remove the daily urea at a rate of 14.2 grams. So here's a summary of the device performance. In 24 hours, we have removed 14.2 grams of urea using active electrode area of 1,810 centimeter square. We have also done a lot of simulations to optimize the urea diffusion to the electrode. What you can see here in figure A is the distribution of velocity over the spacer or the channel. The y-axis direction is the flow direction. The y-axis direction, that's the thickness of the device. As you can see along the x-axis direction in the center, the flow field is very uniform. That's what we want. In figure B, you can see that's the urea concentration being plotted. If you look at the green color of the scale bar, that's 5 millimolar. It gradually extends into the center of the channel as we go down the x-axis. So this urea concentration profile generally follows the fixed second law of distribution, which tells us that still the urea diffusion rate is limiting the urea decomposition rate, which tells us that we could still enhance the urea decomposition rate by optimizing the flow pattern. However, when we transfer the urea, this device into spent dialysate, we found something different. On the top row is the urea decomposition measured in a simple saline solution. It's 0.15 molar sodium chloride. The urea decomposition measured in here is 0.29. That's milligrams per centimeter square per hour. And but when we switched to human spent dialysate, we found that urea decomposition rates dropped very significantly to 0.09. At the same time, there's an increase in glucose decomposition rate to 0.087. So that's a 2/3 reduction in urea decomposition rate, and presumably the photocurrents that were originally used for urea decomposition, they ended up decomposing the glucose. And besides glucose, there are other interference from in the blood serum, and we listed all the uremic retention solutes that are removed in the typical thrice weekly dialysis session here. The uremic retention solutes means uremic toxins plus many nutrients. For example, here number 1 is the glucose, followed by urea. Then there's some amino acids here. If we simply look at the top 20 uremic retention solutes, their total mass removal is 64.3 grams. If we extend that list to the top 50, the total mass removal will be 67.6 grams. And out of that, we only need to remove 23.6 grams of urea. So all these uremic retention solutes, they will be there to reduce the urea removal efficiency. We put the molecular weight of all the retention solutes in the context of molecular weight cutoff of different membranes. We get this figure. Nowadays, most people are interested in high flux dialysis, and people are trying to get precise control of the pore size so that the removal of the large molecules will be easier. However, most of the retention solutes are in this low molecular range, and we have to study the properties of membranes such as reverse osmosis and nanofiltration to get a better control of the filtration of these small molecules. And we have a very good small molecule selective membranes. We imagine we can have a device working like this. First, we have a hemodialyzer interfacing with the patient, where all the large and small molecules will end up in the yellow dialysis loop. The non-urea urine toxins will be removed by these activated carbon layer. And going to the right, we have the forward osmosis cartridge, which is urea-selective. So the urea and only urea will pass this membrane and end up in the oxidation loop. And the urea would be decomposed into carbon dioxide, nitrogen, and water. And so we found this commercially available aquapowering inside cartridge, and we prepared samples of uremic retention solutes, and we sent the samples to Dr. Anna Scott and Dr. Andy Hufnagel in Seattle Children's Hospital and here at UW Lab Medicine to test their concentrations. And on the left axis, you can see the concentration in log scale. The cyan color is the feed concentration, and in the orange color is the FO, which forward osmosis permeate. In the first species was urea. Before and after the forward osmosis membrane, there was almost no drop in concentration. But going to the second, to number 2, glucose, you can see from the cyan color to the yellow color, there's 3 orders of magnitude drop in concentration. So we dropped the glucose interference to almost 0. And the trend is similar for other retention solutes, such as the amino acids and uremic toxins on the far right. So we have found a very commercially available membrane that's highly selective to urea diffusion, which at the same time cut off glucose diffusion. The other question is that whether this membrane can provide adequate urea flux to make it operable as a portable dialysis device. On the left axis, we have the urea removal rate with units of grams per day. On the bottom axis is the inner fiber flow rate in milliliters per minute. With 10 millimolar of feed urea concentration, we found that at a flow rate of 30 milliliters per minute, we can remove 20 grams of urea. That's more than we want to remove with 15 grams of daily urea removal. We have constructed a device like this. The blue cartridge is the forward osmosis cartridge. On the left, we have the spent dialysate being circulated. And on the right, we have the urea photo decomposition unit. This device we did on a small scale. But if we do the numbers, we found that using real spent dialysate, we can still remove 15 grams of urea in a day using about 2,000 centimeter square of electrode area. So that means we have gets rid of the interference of in urea removal. Here plotted is the cytotoxicity of the samples collected during the 24-hour test. And this is a very complicated figure, so I'll explain this a little more. In figure A is the control. In figure B is the starting samples at time 0. SP is short for spent dialysate. POR is for photo oxidation urea removal unit. AC is activated carbon. That means the samples were treated with the activated carbon. First, if we look at the red box in the SP standalone, that's spent dialysate, standalone spent dialysate, which was not involved in the 24-hour operation. This, the cytotoxicity is plotted. Then in the center is the spent dialysate. That means the sample collected from the 24-hour operation. And it has similar cytotoxicity to the standalone samples. On the right, we have the standalone, the spent dialysate that was treated with the activated carbon. And all these three samples have no significant difference in the cytotoxicity. On the right, however, it's quite different. From the left, we have the POR pre-AC. That means the samples collected in the photo oxidation units that were not treated with activated carbon. That's very cytotoxic. Then after a short activated carbon treatment, the cytotoxicity were almost removed. Similarly, the latter two samples, they were treated either with inline activated carbon or further treated with inline activated carbon and then activated carbon treatment during sample treatment. As you can see in the latter three samples, even with a little bit of activated carbon treatment, the cytotoxicity was mostly removed. And we have this forward osmosis membrane that acts as a barrier for any toxic byproduct. So in comparison, the ready system shown in figure A, we have the patient was interfaced to the hemodialyzer, and there was a dialysate regeneration unit that's centered around the urease enzyme with 2 layers of ion exchange materials. On the right, we have our FO-POR, which is forward osmosis photo oxidation urea removal device. We have in the first stage, we have the hemodialyzer as well that's interfacing with the patient. On the second stage, we have the forward osmosis cartridge, which is urea-selective. And all the uremic toxins will be removed by the activated carbon layer in the dialysate circuit. And the urea alone will be removed in the urea removal circuit. And the advantage of this double-stage device is that first, it protects the POR unit, the green circuit, from interference and from non-urea species, and it will enhance the efficiency of urea removal. Second, it protects the dialysate and patient from the byproducts that's produced in the POR unit. And this urea-selective FO cartridge could potentially benefit other methods of urea removal as well, such as the urease-based methods and sorbent-based methods, all because they will minimize the interference of non-urea retention salutes. So here's the conclusion. First, the POR units can remove urea at therapeutic rates, That's around 15 grams of urea in a day. However, while it's removing urea, it will generate ammonium and potentially other byproducts. Second point is that there is a variety of uremic retention solutes that are being removed together with urea. And urea removal efficiency through photooxidation is reduced under the interference. Then forward osmosis can act as a selective membrane for both urea and any produced byproducts. So it will enhance the urea removal efficiency, and it will enhance the safety of the device. The last point is that the FO-Pour device can safely and effectively remove daily urea generated within the dialysis patient. Lastly, I want to acknowledge many people in my team. First is Dr. Bruce Hines. I was working as a postdoc in his group when most of the work is done. Then Dr. Anna Scott and Dr. Annie Hufnagel helped me quantify the uremic retention solutes in my samples. And many people helped me do the device assembly and the testing. I want to thank Dr. Shen for organizing and coordinating many of that efforts, and Dr. Creason for helping me doing the cytotoxicity study. And listed at the bottom are the directors for Center for Dialysis Innovation and the CDI and Northwest Kidney Centers. I want to thank them for sponsoring this work. And thank you all for listening.
So I'll just add Guozheng and Anna back to the audio line, and we'll get into answering some questions. All right, great. Well, Anna and Guozheng, you ready to answer a few questions then?
Yeah.
Happy to.
Perfect. And as a reminder, if you do have any questions you'd like to submit, you can submit them using the Ask a Question panel. So I can see from the Ask a Question panel that you actually had a few questions, Guozheng and Anna, that you'd like to ask each other. Guozheng, did you want to start with yours to Anna?
Yeah, I would like to know what chemical species other than amino acids are characterized in your lab.
Ooh, so beyond amino acids, I think one of our other highest volume tests with pretty high accuracy and precision is measurement of acyl carnitines. So these are medium and short chain free fatty acids that are conjugated to a carnitine molecule for transport in and out of cells, primarily heading to the mitochondria for ATP production. I'd say from a quantitation standpoint, that's our other big test. Actually, when I was wondering if you might be referring to other chemicals we can see on the HPLC analyzer. And so I want to just touch on this briefly. We can see a variety of medications, mostly antibiotics. Quantification of these gets a little bit hairier because we don't have calibration curves established, but that there is always the possibility for a research collaboration where if there's a compound of interest that might be detected or that I know is detected in one of our methods but is not routinely quantified as part of clinical care or management, maybe because it's not relevant or there's other things that are more interesting for, again, a management perspective, that doesn't mean it's not possible to set something up. So what I've really enjoyed about the biochemical genetics laboratory is our ability to kind of adapt and sometimes even react to specific questions, even that happen for our patients. Because we deal with rare disease, every patient can have kind of a different story going on. So sometimes even if we can't report a hard number, we can do essentially side experiments, a slightly different extraction of a patient sample to try to understand what's happening. I'm thinking of an example where there was a very rare enzyme defect that created amino acid dimers, dipeptides, that we didn't have calibration curves for, but knowing having a much better clinical question that actually came from exome results, we were able to do this really intensive acid hydrolysis treatment of the patient sample and break apart all of those dipeptides to turn them back into free amino acids, which we then could quantify. And that process was basically able to confirm the diagnosis. So we have some room to play, which is a lot of fun.
Mm-hmm.
Fantastic, great answer. And I just wanted to follow that up with another question here. If there are groups here that want to contact you to enter into a collaborative partnership, how might they go about doing that?
Sure. Well, for me specifically, best way is to reach out by email. I think some of my contact information is publicly available through the outward-facing Seattle Children's website. But I'm not the only biochemical geneticist, and I like to think I was able to highlight that even if free amino acids are not necessarily relevant to your research, there may be clinical laboratory testing that could be helpful. And so I would encourage anyone who has need of high precision, high accuracy quantification of analytes to check out the test menus of a local institution. Many core lab functions. So Dr. Andy Hoofnagel is over at the University of Washington, and I believe he offered like glucose quantification, calcium quantification. Like there's all kinds of stuff that is measured to monitor patients' electrolytes and physiology in critical illness. This could be really valuable resources. And again, we have to spend a lot of time and energy making measurements like these reliable and consistent. So I would encourage research labs to use us to your advantage. Reach out to a local hospital laboratory. Oftentimes there's contact information on the web. And even getting into a client services system, you may be able to get rerouted to their clinical chemist or somebody in the laboratory who could connect you for establishing a research collaboration. So there should be emails, phone numbers, reach out. We love to share what we can do and further research to help patients.
Fantastic. Yeah, some really great points there. And we can also include Dr. Scott and Dr. Shao's contact information in follow-up emails after the webinar. So in case you want to reach out, we'll provide those. And I also thought that you had a few questions for Guozheng. Do you want to ask those now?
Absolutely. So Dr. Shao, I was curious to see what are the next steps, because your data here is so promising. What's going to happen to get your device ready for actual use for patients? What's next?
We are. Thank you for that question. We are doing bovine blood test for 24 hours, and actually much longer than 24 hours. And we're testing that for a few days. The purpose is to, number one, to confirm the urea removal rate. Number two is to confirm the safety of the device. But that's also a more complicated scenario because coagulations and quantifications of the retention solute. And still another point is that to further enhance the efficiency in terms of urea removal, we have shown that through the simulation that urea diffusion to the electrode is limiting its removal rate. And we want to optimize our flow pattern and increase the efficiency, the rate of urea removal, and shrink our device dimension and weight.
Yeah, fantastic.
Okay, that all makes sense to make it more portable. Anyway, go ahead, Liam. I'll let you manage.
Yeah.
All right, so another question here. This one, Dr. Scott, this one's for you. So amino acid analysis using ion exchange and anhydride detection is a well-established technique developed in the 50s. What is your view on the advances of newer technologies such as LCMS for amino acid analysis? How do they compare with classical AAA analysis?
Right. So I kind of alluded to it, and everybody has, I think, there's differing opinions in the field. LC mass spec is potentially very powerful. You know, you can make your library of targeted amino acids to detect, but an LC mass spec is best when you have, in fact, that targeted library. They're not as good yet, particularly for quantification, for untargeted approaches. And the instruments themselves are cranky. They take a lot of love to keep them going. They take a lot of daily, weekly, monthly maintenance. Sensitivity, they can be extremely sensitive. So I'm thinking about quantification of arginosuccinic acid, which is a very rare amino acid, but clinically very significant, even at small levels. So I think a mass spec for amino acid analysis is much better at detecting arginosuccinic acid, but the technical expertise needed to both develop the initial method and keep it going is something that a lot of smaller labs, particularly like my lab, really just, we don't have the resources. We don't have the upfront budget or the personnel to keep that kind of an instrument running. Some of our colleagues at the big reference labs, so I'm thinking Mayo, ARUP, Quest, and LabCorp, where they get hundreds to thousands of samples a week, that's a really different scale than my team, where we get 10 to 20 a day. They can then justify the upfront cost. They also have a much bigger lab support team to keep the LCMSs going. But as I guess, kind of as I alluded to with the previous example of the dipeptides, a tandem mass spec will miss weird stuff going on that's not part of its program. So there's actually a wonderful paper out there about how repeated analysis by tandem mass spec of free amino acids failed to diagnose prolidase deficiency because dipeptides are not built into these libraries. It's very rare to see them. And so in that scenario, the classic HPLC ion exchange and anhydride amino acid analyzers are the better instrument. Our cost per sample once the instrument is set up, I'd say is actually a little bit higher, but the upfront cost of the mass spec is pretty daunting. So each have their pros and cons, and I think it really, the technology that is appropriate depends on the needs of the laboratory and the resources of the laboratory.
Fantastic. Yeah, thanks so much for sharing. I think we'll try to squeeze in maybe two more questions here. Guozheng, this one is for you. So thinking about inborn errors of metabolism, there are patients where dialysis is needed to remove other molecules like ammonia or leucine. Do you think it would be possible to make other two-stage devices to enable selective removal of compounds that could be patient or disease specific and minimize the loss of those beneficial metabolites?
I like that question. There's a lot of opportunities there. I think it's definitely possible when we develop membranes with new properties that's selective to a new class of molecules. And on the other hand, I think the ready system could potentially be readily useful here, as there is already ion exchange materials. We can actually remove the urease from the ready system. We can just leave the ion exchange materials there, which will remove ammonia, and amino acids, and other ions as well. So then obviously you need to back infuse the other amino acids and electrolytes. So that's already the ready system. It's by removing urease from that system, it already works, and by introducing new membranes, potentially the two-stage system that we are developing could also work.
Fantastic. A few maybe short questions that we have left. Guozheng, this is another one for you. What detection levels do you require for the dialysis investigation in terms of picomoles?
It's complicated because if you really go to the picomolar region, the list of molecules you can detect can be super long, and we don't want to burden ourselves with quantification of all the chemicals at this stage. And we're just interested in the at most the top 60 or 70 uremic retention solutes. So they are mostly above the millimolar regime. Some of the chemicals are around maybe 0.1 millimolar. Yeah, but we are not going much more than that regime at this stage. There's one exception, that's ammonium ion, which we need to pay special attention to because ammonium ion is toxic to the patient above 50 micromolar. Yeah, hope that answers your question.
Yeah, definitely. Well, thank you so much, Guozheng and Anna, for the really fantastic insights today. It's been a real pleasure having you with us.
Thank you for organizing it, Liam.
Thank you.
Yeah, absolutely. My pleasure. Big thanks also, of course, to the audience here for participating. And last but not least, we'd like to also thank Biochrome for sponsoring this event. We now invite you to take your time to submit your feedback on this survey. We do read all of your questions and comments and really appreciate any input you can provide. If you still have a question, it's also not too late. You can still submit it using the Ask a Question panel, and we will be forwarding it on to the speakers following the event. Again, if you're interested in learning more about the research or the technology presented today, we have added some really great publications and web resources in the resources tab. And so in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.